Can CRISPR turn a rock pigeon into a passenger pigeon-like bird that reshapes forests? ReShape, Revive and Restore, has run multi-year gene editing work in pigeons as a model. Receipts first, verdict later. Is a proxy enough, or is this cosplay biology? The claim sells a simple dream. Swap genes into a common pigeon and abracadabra, we get flocks that darken the sky and kick forests back into the dynamic disturbance cycles of the 19th century. The actual lift is closer to a moonshot made of many small stubborn steps. First, you need the right blueprint, ancient DNA from museum skins to map what made the passenger pigeon a high-density boom and bust specialist. Then you need a chassis, a living bird you can breed, handle, and edit in real labs. That's the rock pigeon model, chosen because it's tractable for avian gene editing and husbandry, not because it's the closest relative. Next, you must stack edits that matter for traits like schooling, migration cues, body plan, and social hormones, edits that affect behaviour and ecology, not just feather colour. Finally, the lift demands germline transmission, so the traits pass to offspring, plus a path to scale from a few edited founders to real populations without inbreeding collapse. None of that is one giant lever. It's a rack of fiddly dials you turn for years. Here's the state of play. Revive and Restore's Great Passenger Pigeon comeback established the model species pipeline in pigeons years ago and continues a multi-year research and development program. The core idea is avian germline editing, getting changes into sperm and eggs so traits inherit, and that means primordial germ cells. Isolate them, culture them, edit them, and prove they make functional gametes. In birds, this is notoriously hard, which is why so much progress reporting talks about PGC culture, chimeras, and proof of concept matings. Parallel projects in conservation, like the pink pigeon work, show how PGC tech can reintroduce genetic variation into threatened birds. The same toolkit underpins any passenger pigeon-like proxy. But note the gap, there is still no true passenger pigeon and no published flock of functionality equivalent PP-like birds interacting with forests. The work remains model stage and methods heavy, promising but not a field test of the headline claim. Let's be real about the animals involved. Proxies aren't born from press releases, they're born from other birds. Avian de-extinction replaces cloning with germline hacks, so you're producing edited germ cells, inserting them into host embryos, and asking a surrogate species to do the hard biological lift from egg to hatchling. That raises non-trivial welfare questions. How many host embryos get used to dial in the technique? What's the survival curve? And what's the plan when early generations express off-target traits that make them poor parents or maladapted juveniles? Scaling is its own ethical slope. A charismatic project will face pressure to go faster, but welfare debt compounds when you try to jump from a handful of chimeric breeders to a managed population with enough genetic breadth to avoid bottlenecks. Add captive rearing stress, imprinting quirks, and the risk that edited lines selected for lab convenience rather than wild competence. If the endpoint is ecological function, branch-breaking flock density, and disturbance pulses, you're selecting for behaviours that may be stressful to individuals and landscapes. None of this is an automatic veto, but is a tab you must show. Technology can open the door. Animal welfare must decide how far we walk through it. Even if a proxy hatches cleanly, you're not releasing a museum exhibit, you're releasing a force. The historic function was disturbance on a continental scale. Branches snapped, seed range shuffled, Mars cycles jolted. Today's eastern forests aren't the same mosaic of land use, predators, and disease. Where exactly do you rehearse billion bird physics with 100 edited starters? If the answer is carefully chosen sites with intense monitoring, you've admitted the hidden cost. Long, expensive aftercare. That means telemetry on every cohort, forest plot experiments to catch unintended shifts, disease screening to prevent novel spillovers, and governance so a regional nuisance doesn't bloom from good intentions. There are smart baby steps, forestry proxies that simulate disturbance without birds, but those pilots also stir debate about whether we're restoring processes or playing Jurassic landscaping. Before a single release, show a budget for a decade, name the liability holder, and specify your emergency offerings if behaviour trends the wrong way. If we can't carry the aftercare, we shouldn't start the care. 2 bottlenecks matter most. First is durable germline control. You need a reliable recipe to culture, edit, and transmit pigeon PGCs at scale with predictable inheritance, minimal off-targets, and no silent fertility tax that appears in F2 or F3. That's getting better across avian science, but better is not bankable, and every species throws its own biochemical tantrums. Second is trait stack validation. Editing one marker is a lab win. Editing suites of loci for behaviour, physiology, and morphology, and showing emergent flock dynamics moves you into hard biological theatre. You must prove the proxy doesn't just look right, it acts right under wild-type social densities. Add the translation layer, taking techniques validated in chicken or pigeon lines and porting them into the exact lineages you plan to breed as founders. Each translation step risks delay until a program shows germline edits flowing into multi-generation pedigrees with field-relevant traits. The hype stays ahead of the hatch. Solid progress, yes. Shovel-ready ecology? Not yet. That gap is where timelines go to die and where sober projects either slow down or finally publish the receipts everyone's waiting for. Here's the argument that divides the room. Proponents say a functional proxy earns its keep by restoring lost processes, not lost genomes. If you can recreate enough passenger pigeon-like behaviour to kick forests into richer dynamics, who cares whether every letter of DNA matches Martha's? Skeptics counter that function isn't a costume you throw on a rock pigeon. It's an evolved package: density-dependent breeding, nomadism, predator swamping, social learning, and boom phase physiology. Miss a few ingredients and you don't get a landscape engineer, you get an odd pigeon with a brand. Meanwhile, conservation triage asks a brutal question. For the same money and media oxygen, could we harden habitats for living birds and restore disturbance through forestry that we control and reverse? That's not romance, that's return on effort. If proxies become a magnet for attention while living species slip, we've performed ecological cosplay while real diversity thins. The only honest reconciliation is receipts, field-scale trials that show measurable uplift against controls, and a funding pie that doesn't rob the present to chase a curated past. Verdict: veto for now. The tech arc is trending the right direction, and avian germline editing has matured from maybe to sometimes. But this claim isn't a bird, it's a brochure. There is no published passenger pigeon-like flock, no field demonstrated functional disturbance, and no transparent path from a model pigeon line to region-scale ecology. Add unresolved welfare and long-tail aftercare, and the program's centre of gravity is still lab-side. Changed my mind with receipts. Peer-reviewed germline transmission in pigeons with multi-trait stacks, F2 stability, and pre-registered forest trials with independent oversight also show a habitat governance plan that names who pays and who stops the show. Until then, fund the enabling science and publish like your credibility depends on it, because it does. When those receipts arrive, this can move to a cautious green light pilot. Today, it's no to gene editing forests back to life, and a yes to publishing the work that would make a careful pilot worth the risk. If function is the promise, prove it where the risk is lower.